many different animal groups have been looked at for their investigated for their role of hosts of emerging zoonotic viruses. Rodents and bats have been amongst the forefront of these animal groups, specifically because they're very species rich and they host a large number of viral species, and therefore a high proportion of their viruses are zoonotic. Both of these animal groups, though, if not all of these animal orders, should be included in wildlife surveillance activities, and not only specifically targeted groups, because they all are actually quite important. If we specifically look at some of these virus groups, again, some have a high proportion of zoonotic or potentially zoonotic viruses. There are many different types of viruses that are hosted by bats. Some are seemingly only part of the virome of the bats, so the normal composition of viruses that are hosted by different animals, and others are definite zoonotic viruses. Then you get some viruses that have a zoonotic potential. When they're characterized after being identified in bats, they fall into genera that are known to host animal and human viruses, so they are related to these viruses, so their zoonotic potential are unknown. There are very diverse viral families that have been identified from bats. This is largely in the last 80 years, from targeted surveillance activities, arbovirus research, metagenomics, specific isolations to see what can be grown in cell culture. Then, so all of these families over here and the African icons indicate the viruses that have been identified from African bats as well. So if we actually only look at the direct zoonotic viruses or the indirect zoonotic viruses, we can actually reduce this list to just a few viral families, including the Filoviridae, Paramyxoviridae, the Rhabdoviridae, specifically the Lissaviruses, and then the Rheoviridae, which you can notice there's no African icon, so no zoonotic Rheoviruses have been reported from African bats. And then I highlighted the coronaviruses because this is a debatable topic of how zoonotic these viruses can be, which we will get to in a little bit. So I just want to highlight some of the other viral families, but today's talk will really be focused on coronaviruses. So the Filoviridae are, of course, filoviruses like Marburg and Ebola. They cause severe infections with possible hemorrhagic fever and high fatality rates. Specifically, these maps over here in gray shows the countries from which RNA have been reported from bats. The diagonal lines also show human cases, and the colored dots throughout the map shows the distribution of the bat species from which these viruses have been identified in. You'll notice with Marburg and Ebola, there have been both reports from bats and people. And then for Marburg virus, the most important one I highlighted there in blue is the Egyptian rosette bat, or the Rosetus aegyptiacus bat, because this is the known reservoir host for Marburg virus. For Ebola virus, it's not quite as clean cut. There have been multiple frugivorous bats that have been implicated as possible hosts of Ebola virus, but most of the evidence for reservoirs of Ebola virus come from serological evidence and not virus isolation. So the true reservoir for Ebola is still under investigation. But for years, because of the serological evidence, it was believed that fruit bats must be the hosts of Ebola viruses, particularly also due to its close relationship with Marburg virus. But the more people have started looking at insectivorous bat species, the more they've discovered that there is a completely unknown repertoire of filoviruses potentially in insectivorous bats. For example, there's Iloviovirus from Miniopterus bats in Spain, and from Africa, Bombali virus has recently been reported from molluscid species. So general filovirus surveillance should really be prioritized in all species and not just those assumed that could potentially be the hosts of these viruses. In terms of paramyxoviruses, for those who are unfamiliar with these viruses, they cause largely respiratory infections and infections of the central nervous system. Some examples globally include measles and mumps. Specific zoonotic paramyxoviruses include Hendra and Nipah viruses, which have already been mentioned today. They're mostly prevalent in Southeast Asia and Australia, but similar viruses have been reported from African fruit bats. So these viruses are also of considerable importance for public and veterinary health, given their association with very high morbidity and mortality in their hosts, whether they're humans or animals, as well as a lack of effective post-exposure treatment. These viruses are also transmissible via urine and to a lesser extent saliva, which have been detected from partially eaten fruit, so increasing possible routes of exposure. Antibodies against Henipa-related viruses have also been detected from pigs in Ghana and from people in Cameroon, which shows that there is a link between exposure continuing between livestock, and pigs can act as a possible intermediate host, and that there's definite exposure happening with people. In addition to the Henipa virus diversity, there have also been African bats that, from which viruses related to human mumps virus has been identified, human parainfluenza viruses, and then also the zoonotic bat virus, the Suga virus, from which only one human case has been reported, but these viruses also circulate again in the Egyptian Rosetus aegyptiacus. All right, and then lyssaviruses. Of course, these are the rabies and rabies-related viruses. Just to highlight, there is no rabies virus in bats in Africa, but there is a similar viral diversity in bats in Africa that is capable of causing rabies-like disease in mammals. There are multiple lyssavirus species from Europe, Asia, and Africa, but 4 species in African bats, namely Duvinaga, Shimoni, Lagos bat, and Matlo lyssavirus. Duvinaga is associated with human fatalities, and the transmission occurs through either direct contact with infected saliva or on open wounds or mucosal membranes from a bite with an infected animal. The host species for some of these lyssaviruses are not quite as clear-cut as some of the others. For Matlo virus, it's Miniopterus. This is a new virus that's been described. And for Shimoni bat virus, lyssavirus, it's Macronectaries. But for Lagos bat, it actually has a wide distribution amongst different bat species. And then for Duvinaga, it's a bit conflicting. It could be Miniopterus, it could be Nectaries. Some of the data is suggesting one over the other. But in overall, serological studies are generally more successful for active lyssavirus surveillance because viral RNA is generally only in the animals showing symptoms of disease. So active surveillance have really low returns on investments. If you sample a whole population of healthy bats, your positivity rate is very low. So passive surveillance is really how lyssavirus research has continued by direct submissions to reference labs or research groups. And then we get to coronaviruses as zoonoses. So this is a bit of a debatable topic because unlike filoviruses or lyssaviruses, if you come into contact with a corona-infected bat, your chances of getting a coronavirus infection is quite low. It would be better to look at coronavirus infection from bats as they require a repeat opportunity, they require a repeat exposure, adaptation to new hosts, potentially shared receptors. It's not as high impact as filoviruses or lyssaviruses as examples. There are 4 endemic human coronaviruses in the human population with a global distribution. Now there are 4 newly emerged coronaviruses since 2002, which one might be becoming endemic. So the general trend with all of these new viruses are that they emerged into a naive population. And the same mode of emergence where exposure and adaptation is required has happened 4 times in the last 22 years alone, which shows that even though these viruses are not a direct zoonotic risk, there is still a zoonotic risk, which is why I refer to them as indirect zoonoses. And each of these new viruses has in some way been associated with either related viruses or possible progenitor viruses in bat species. Even if you look at the previously known endemic viruses, each of them has an association with the zoonotic origin. If we look at human coronavirus OC43, it's potentially derived from bovine coronaviruses years ago. Human coronavirus HKU1 is suggested to have emerged from a whole large diversity of rodent coronaviruses that's been well characterized in Asia. And then both the alphacoronaviruses, 229E and NL63, has been associated with bat species, specifically from Africa. So just to give some terminology that you'll hear a lot throughout the talk today, there are 4 genera of coronaviruses. The alphas and the betas are associated with mammals, and the gammas and the deltas mostly with birds, but they have also had sporadic detections from other mammal species. And the alphas and the betas are further divided into several subgenera. So, for example, if I say Sarbecoviruses, I'm specifically referring to the Sarbecovirus subgenus, of which there's one species, the severe acute respiratory syndrome related coronavirus, and this refers to SARS-1 and 2, and all the SARS-related bat viruses, as well as some of the viruses that occur in other animals. So quite a large diversity within one species. And as you will look at these subgenera, you'll notice a lot of little bat icons, which shows that there's a lot of diversity that is unique to the bats. And in some situations, there's diversity that occurs in bats and other animals. And then, of course, people as well. So just to give a quick recap of global coronavirus surveillance that's happened since 2005, it is currently believed that bats are the ancestral hosts of coronaviruses and that they host the genetic diversity of alpha and beta coronaviruses, which later then led to diversification in other animals. There is a general host genus specificity that has been reported from bats, where you can find related bat species in related bat hosts, related viral species and related bat hosts that seems to be geographically independent. So you can probably find similar viruses when you sample the same genus of bats in Africa, Europe, and in Asia. And then also there's frequent cross-species transmissions between bats. Sometimes it leads to adaptation and a specification in a new host species, and sometimes it's a dead-end infection. And when you detect this through surveillance, you don't really know if the virus is actively replicating in this new host or not. There's also a high level of recombination between coronaviruses and bats, between most coronaviruses. And then lastly, these viruses also show a gastrointestinal tropism from bats, which makes collection of sample types, fecal or rectal swabs, actually relatively quite easy. So if we look at Sarbecoviruses, like I just mentioned, that there is a general host genus specificity to the hosts. So when you're targeting specific viral groups, it actually reduces the amount of bat species or bat genera that you would have to target if you were looking for something specific. So the Sarbecoviruses are associated with the Rhinolophus genus, or the horseshoe bats. Whether they occur in Asia or in Europe or in Africa, wherever these bat species are distributed, these viruses have been identified. So we know that there is a lot of genetically related diversity that's related to SARS-1 and 2 occurring in bats throughout this distribution. There's actually 5 lineages that have been recorded. In terms of intermediate host, the Himalayan palm civet was suggested to be the possible intermediate host of the original SARS outbreak. For SARS-2, multiple animal species have been suggested, but it has also been suggested that these viruses were capable potentially of direct transmission from bats to people. Like I've said, throughout the distribution of these hosts, however, there are very few species that overlap between continents. So there isn't just one species that occurs throughout that you could target for Sarbecoviruses. In Africa, there are 14 Rhinolophus species currently. And from 2 of these species have Sarbecoviruses been reported. And you'll see their distribution is quite wide, but only a few countries have actually reported Sarbecoviruses, from Uganda, Rwanda, Kenya, and now in South Africa. So I'm fairly certain that some of this in-between countries would also be reporting Sarbecoviruses if there was sufficient surveillance. I just want to highlight this subgenus as well. So this is related to Sarbecoviruses, the Hibecoviruses. The HI comes from their host genus to which they are associated, which is Hibecoviruses, as well as the related Merbecoviruses genus. So these viruses are not Sarbecoviruses. They are a sister taxa to Sarbecoviruses. But I've seen in a lot of the literature, people refer to them as being SARS-related, but they are definitely not. That's why they're in a different subgenus. There is some level of either habitat or ecological sharing between Rhinolophus and Hibecoviruses bats, because you will also find viral sharing between these genera. Sometimes you might find a Sarbecovirus in a Hibecoviruses bat, but this is rare and not often encountered. And sometimes you'll find Hibecoviruses in Rhinolophus. These viruses are also not known to be zoonotic. Their potential is unknown. If we look at MERS, the situation is a little less clean-cut. MERS emerged on the Arabian Peninsula in 2012. The virus is believed to have been spread from dromedary camels, which is considered the reservoir host, to people, and there is also limited person-to-person spread, which is mostly associated with nosocomial infections. So coronaviruses similar to MERS have been identified from bat species in Asia and Africa, both before MERS actually emerged in people and as well as after. So dromedary camels have been, like I said, they are the hosts of MERS viruses, and it's believed that African bat species carrying this MERS diversity could potentially have spilled some of this viral diversity into camels, and they have been circulating for quite a few years. Support for the circulation and adaptation of these viruses in camels comes from both serological evidence from sera that's been collected from camels as early as the 1980s, shows that there are antibodies against MERS in the camels. And there are also various different viral lineages occurring in dromedary camels, whether they are sampled in Africa or in the Middle East, which show that there is some adaptation locally. So the MERS virus is present in bats. If you compare them to the ones in camels and people, they share about 85% nucleotide identity across the genome. And in some of the more important or conserved gene areas, it's sufficient to classify them all as one viral species. The area that is the most different of these genomes is the spike gene, which is responsible for receptor recognition and attachment, which shows that the spike genes really had to undergo the most adaptation and change to move from bats to camels, and then this change was facilitated more easier to people. So this host diversity from that occurs in bats is a bit more widespread than just Rhinolophus and SARS. It actually, you can associate it with the family Vespertilionidae, from which there are now multiple genera associated with MERS-related viruses, specifically Lophotis campensis and Pipistrellus hesperidis. There has recently been taxonomic changes to some of these hosts, so we could actually consider some other hosts that could also potentially be hosts of MERS-related viruses, specifically Neuromecia and Afronicteres, just to mention a few. And this actually shows how important it is to have correct species identification of viral hosts, because it will have implications for risk management as well. So as you can see on the map, very few countries are gray, so very few reports of MERS-related viruses in bats. But you'll also see the distribution of these bats are very abundant and very widespread throughout this map. So I think surveillance is potentially the problem in that case. Then I just want to highlight some of the human alphacoronaviruses. So human coronavirus 229E is also associated with the Hibecoviruses genus. In this case, these are the blue dots on the map. So there's quite a large diversity of human coronavirus 229E related viruses that are circulating in Hibecoviruses, and also quite widespread throughout Africa. They've been reported from several African countries. It's as shown by these diagonal lines. The gray areas show where both of these viruses have been identified, and this one is the only one from Trianops afro alone. So NL63 has been associated with the Trionops afro species. As you can see, not very widely distributed throughout Africa. And therefore, likely that's why the detections are low. If you look at some of the viral diversity, along with the human and Hibecoviruses 229E related viruses, there's also a diversity in camels and alpacas that has been detected, which shows that maybe in the history, during the emergence of human coronavirus 229E, alpacas or camels or some camelid species may have been involved in the transmission to people. All right, so I just want to highlight some other undescribed diversity. There is a large diversity of bat coronaviruses, and for most of them, they have an unknown zoonotic potential. So I've mentioned the ones at the top, but I really want to highlight some of the others. These viruses occur in very abundant hosts that form very dense populations. If we look specifically at the Rosetus genus, Miniopterus genus, and then diverse molluscid species, these two insectivorous species host a large diversity of alphacoronaviruses that are quite poorly studied. And then I just want to also mention the Merbecoviruses, which is a subgenus in the beta coronavirus genus. So these Merbecoviruses occur throughout the distribution of their hosts, which are old world fruit bats. So wherever these bats occur, these viruses occur, and in quite high dense prevalences as well, with positivity rates of between 5 to 30%, which is quite high for coronaviruses. So basically, we tried to take a lot of, we collected all the sequences that we could from surveillance in wildlife, specifically bats and all the animals we could find, and we threw them together into a phylogenetic tree. I know you can't see this. It's not the point that you do see this because there are too many sequences in this, but the area in the sequences that's blocked in pink actually indicates the Merbecovirus diversity. So when you compare this to the rest of the tree, 3/4 of the tree is made up of Merbecoviruses, which shows that these viruses are very commonly encountered. That's why there are so many sequences of them, and there's quite a large diversity as well. But very sadly, these viruses are poorly understood, they're poorly studied because people don't see them as a zoonotic risk, and they look at some of the other viruses, and this goes unstudied. Same thing for the alphacoronaviruses. I know you can't see this, but this shows a large diversity of African alphacoronaviruses from bats and other animals, mostly from bats. And as you can see here on the side, there are mostly unclassified viruses associated with bats, and sometimes they're also associated with other animal species, which just highlights that there's a large diversity of bat coronaviruses that are understudied. So there are also bat hosts that have been identified as hosts of other viral species, and I just want to point out one of them. The Egyptian fruit bat, Rosetus egyptiacus again, has been associated as a host for many different viral families, very different viral species, Marburg, paramyxoviruses, lyssaviruses, etc. So it just goes to show that you shouldn't narrow down your surveillance focus to only one specific virus. If you're already going through all the effort of collecting samples, collect for many different viral species that you can get the most return for your investment. So, okay. So in terms of African coronavirus surveillance, we wanted to construct, we wanted to see what has been done, where are the research gaps. So we summarized all the literature that we could find, keeping in mind this is published research. So if you know of something that we missed, please come and tell me at lunch or whenever. We really want to include it. So the map shows increased research effort, or rather research output, because we know that there is unpublished research that would probably belong on this map. But for now, this is the published research. So, so the more blue the areas of the map are, the more publications have been reported. These little icons also indicate the diversity that has been reported from these countries. You'll notice that there are very few red and orange little diamonds, which shows the Sarbeco and the Merbeco diversity. there are a lot of these little yellow triangles, which shows unclassified diversity, and a lot of these little purple squares, which shows the Merbecoviruses. All right, so when we were doing that literature survey, we also wanted to see what was the sampling effort and of the bat species that have been targeted, and are there any trends that we could determine. So you'll notice about 28,000 bats have been tested for coronaviruses. Now, some of the publications didn't make it easy to get to this total. So this is at least 28,000 bats have been sampled, though likely a lot more have been. We are continually updating this map with an overall positivity of about 6.7%, if I'm rounding up. You'll also notice some of the bat families. So these list the bat families and then the different coronavirus subgenera. You'll also notice some of the bat families are quite undersampled, as I've indicated them in gray. I want to highlight this bat family. With not insignificant sampling effort, it still has had no coronavirus positives, which is quite interesting. Then in terms of Sarbecoviruses, overall, you'll see that this accounts for less than 1% Sarbecoviruses from bats. But if you specifically look at the host that they are supposed to be in, it's just over 2%. So considering all of this surveillance effort, only about 740 Rhinolophus bats have been surveyed for coronaviruses in the whole of Africa. So this likely also indicates that there is the prevalence of Sarbecoviruses in bats is probably underestimated. When we increase surveillance, this number might go up, it might not. So overall, what was most detected have been Merbecoviruses from fruit bats, a very high prevalence of the NL63 related diversity in Trienops bats. And then mostly it's been unclassified viruses, and usually this is unclassified alphacoronaviruses. So most of the research done in Africa has been cross-sectional studies with limited longitudinal surveillance, reporting on diversity, and very few actually characterizing the viruses or trying to understand the ecology. These were preliminary research papers, so it makes sense. However, if you want to actually understand how these viruses are maintained in their hosts, you need to look at the epidemiological or ecological factors associated with maintenance. So you would have to start looking at things like excretion timing or the duration of excretion, the demographics of the bats that are most infected with the viruses. Is there viral clearance? How long does immunity last? All of these viruses, all of these drivers that could lead to persistence potentially of viruses in bats or not. And once you understand the factors involved, you can then determine what is associated for risk, essentially. So of all these studies, very few have looked at these factors. And those that do start by looking at things like reproductive stages, for example, mating, and the associated hormonal changes may make bats more susceptible to infection. Or, for example, if there's a birth pulse and a large increase of immunologically naive individuals, are they susceptible to infection, and is that how these viruses are maintained in the population? So there are some agreements between studies and some disagreements. First is the life stage, and definitely young bats are more likely or more associated to be positive with coronaviruses, particularly if they have been recently weaned, by 3 times more likely from one modeling study. Between gender or reproductive stage, there's been some disagreement. Some studies report that lactating females have a higher chance of being positive. Other studies report on males. The largest discrepancy between studies has been seasonality, where some studies report the wet season versus the dry season is better. The other studies report it the other way around. Ultimately, all of these are generalizations. If you consider that bats occupy diverse niches, they have different reproductive cycles, they have different habitats, different roosting behavior, different feeding, different population sizes. All of these things would contribute to how viruses are maintained. So classifying them all together is likely going to result in biased conclusions. So seasonality being a disagreement factor is probably as a result of the different species in these studies and their reproductive life stages. So if young bats are important, when are the young bats present in the environment, essentially? So all of these factors are quite important when you are trying to investigate or trying to understand how these viruses are being maintained in a population. So we also wanted to see what was the effort of wildlife surveillance for coronaviruses. And again, we did a literature survey, very few publications on this. You will see that this table, which summarizes the data, shows quite a large number of animals that has been sampled. When you can compare this to the bat, 28,000 bats versus nearly 40,000 animals that have been surveyed. But again, this large number is across multiple animal orders, many animal species. As you can see, a lot of this is very undersampled. I just want to highlight, even though, and a lot of this data is from the PREDICT surveillance, which is also opportunistic, not repeat sampling, but all of this is also nucleic acid surveillance. So your timing with nucleic acid surveillance has to be when the animals are infected. Otherwise, you'll get a lot of negative samples, which makes nucleic acid surveillance for wildlife quite tricky and really should be paired with serological surveillance. So I want to highlight some of the positives that have been found. These 14 non-human primates are actually gorillas from the Ivory Coast. And it's actually a case of reverse zoonosis, where the gorillas were infected with human coronavirus, OC43, from a caretaker at the sanctuary. So a case of reverse zoonosis. So the majority of these positives come from the rodent and the shrew diversity that have been sampled. And we know that there is quite a large diversity of these viruses in this animal group. And also interesting, African palm civets have also yielded novel coronaviruses. So definitely small carnivores are a host to keep an eye on. So many conditions that need to be met for there are many conditions that need to be met for coronaviruses to spill over, and we don't really know what the precise mechanisms are. We can postulate that there are opportunities for spillover can occur when people or animals disrupt bats in their natural habitats. Each piece of the puzzle can be considered independently, but they are all interconnected. How the viruses infect the bats, how they're being maintained in their populations, what viruses are present in wildlife, and how there is interaction between bats and wildlife. Is there opportunities for recombination between viruses, for example? And then what are the interfaces where people interact with animals and bats, and where are the opportunities for spillover? So all of this you can then combine to look at one large picture. Some of the factors will increase risk, some of the factors will decrease risk, but at the end of the day, it allows you to establish what is the risk of spillover to occur. Is it very low? Is it very imminent? So in terms of planning a surveillance program around bat coronavirus surveillance, it's a very good idea to begin with what has been done before in your region or in your country, so that you will know, okay, what species of bats do we have in our country? Always a good starting point, because that will help you determine what viruses are associated or what viruses you will most likely encounter. This helps you devise your research question, it helps you establish a research setting, and it also helps you plan from there. If we look at South Africa as an example, we will, based on what host bats are in our country, we will likely find SARS-related viruses, MERS-related viruses, and we have a lower chance of finding viruses related to 229E, and we can probably look for NL63 until we're blue in the face, because we don't have the hosts in the country. So just in terms of some of the other intermediate spillover hosts, it's also a good idea when you're planning your research to see what hosts are also in the same area or setting. So we know a lot of animals have their own repertoire of coronaviruses that circulate in these hosts. We also know that some animal species are susceptible to acquiring human coronavirus infections, For example, mink and lions have been associated with infections of SARS coronavirus 2, which were probably picked up from people. And then civets also associate with the original SARS outbreak. I've already mentioned the gorillas and human coronavirus OC43. We also have these animals that's in between. They likely host their own coronaviruses, but have also been implicated to potentially have shared their viruses with people, causing the emergence of new viruses. And then we have unknown hosts that we don't know if they have their own coronavirus diversity, but these are the animals that occur in the areas where surveillance is likely to occur, where spillover is likely to first be seen. So I just want to end off on this slide to show that a lot of really good research has been done in Africa with a lot of really good discoveries, but there is still room for improvement, a lot of blank spaces on the map that we can fill in. And as I've mentioned, a lot of issues that still have to be determined in terms of the maintenance of these viruses. And I just want to thank the following funding agencies.